Hello, everyone. I'm Dr. Arpita. A very warm welcome to my genetics classes. We will talk about epistatic interactions here. I have subdivided this lesson into various parts. This is lesson 1 of epistatic interactions. So let's get started. Before we start our discussion today, we must first discuss what is epistasis. Epistasis can be defined as the interaction of genes that are not alleles, that is, non-allelic interactions. In particular, the suppression of the effect of one such gene by another. Right? So in simple word, you can define it as a masking effect. What is that? The effect of one gene is masked by another gene, and that is non-allelic, right? It is a phenomenon in genetics in which the effect of a gene mutation is dependent on the presence or absence of mutations in one or more other genes, respectively termed modifier genes. Okay, they're termed as modifier genes. Or in other words, the effect of the mutation is dependent on the genetic background in which it appears, right? Now see, before I move into the detailed discussion, I said that it is masking, epistatic interaction is associated with masking the effect of the phenotypic expression of another gene. Students often get confused between these two terms, dominance and epistasis. Dominance, always remember, dominance is intra-allelic gene interaction, intra-allelic gene interaction, whereas epistasis is inter-allelic gene interaction. Here what happens, you see, one allele hides the effect of other allele at the same gene pair, at the same gene pair. Okay, but here you see one gene hides the effect of another gene at different gene loci. This is the basic difference between dominance and epistasis. This is allelic and this is very much non-allelic. Okay. Now we will move on to our main topic today. We will talk about recessive epistasis here. Okay. See, in recessive epistasis, I have just written it out over here. Recessive alleles at one locus, recessive allele at one locus masks the expression of both dominant and recessive alleles at another locus. Okay, this is discussed here with one very important example in plants, that is color of maize grains. 3 colors of maize grains are here. Purple color is governed by gene P and R, that is capital P and capital R, dominant. Red color is governed by capital R, that is also dominant. And white is governed by small r, small p, in homozygous recessive condition. Okay, now look here carefully. See. Concentrate carefully here. Purple flower has got this genetic complement, capital P, capital R, capital R. White has got small p, small r, small r. This is your parental generation. F1 generation yields all purple grains. Complement is what? Capital P, capital R, small r. Now intermating of F1 individuals will give you F2 grains or F2 plants. Here you see, now we will draw the checkerboard to see what happens. Okay, this checkerboard gives you these many number of individuals, right? The checkerboard gives you these many number of individuals. Now we will see which are red ones, which are the red ones, which are the purple ones, and which are the white ones. First, we will look for the purple ones, right? I will only use tick marks of respective color. This is purple. Why? Because as we have discussed here, you see, purple color is governed by capital P, capital R, these two genes. So wherever capital P and capital R are present together, they will all be purple. This is purple. This is also purple. This one is purple. Here in this row, how many purple are there? This one, this one. These two will definitely be of some other color, not purple. Here you see how many purple are there. This one. Then this one. Right? Here in this row, how many? Only this one. Now what about these? Next, we will see how many white grains are there. Okay, white grains, we know this will definitely be white. As we have said, what? Capital P, small r, small p in homozygous recessive condition yields white grain. So this is white. Next comes the twist in the play. This is also white and this is also white and this is also white. Why are these three white? Why are these three white? Here we will have the reason. Small r is recessive to capital R. Small r recessive to capital R, but epistatic to alleles. Epistatic to alleles capital P and small p. This is very, very important. Small r is recessive to capital R, but small r, let me write it, small r epistatic to capital P and small p, right? In F2, all plants with capital P, capital R, see capital P, capital R. If I use purple color here, you will understand better. Capital P, capital R. This complement, this will yield purple grains. Okay. And those with capital R and both small p's. Capital R, see, capital, one second. Capital R and both small p, this will yield red grain color. Red grains. Okay. The epistatic allele R in homozygous condition will produce plants with white grains from small r, capital P and small r, small p genotypes. So we will look for this first, capital R, small p. Here we have capital R, small p. So this will be red. Here we have capital R, small p. This will also be red. Here also we have capital R, small p. This will also be red. Okay. So how many total purple flower we have? We have, see, how many purple flowers? Let's use colors like this. 9 purple flowers we can see. How many white flowers we can see here? Or how many red flowers? We can see 3 red flowers. And how many white we can see here? 4. Okay. So not flowers, sorry, grains. So purple grains is to red grains is to white grains is this 9:3:4. So the typical ratio of dihybrid cross that is 9:3:3:1 gets modified to 9:3:4 here. We will try to write the reason once again here. Okay, these type of interactions should be very clear, very, very clear. The epistatic allele are, carefully see what I do. The epistatic allele are in homozygous condition, in homozygous condition will produce plants with white grains from and this genotypes. Correct. So if this is clear to you, if you can remember this, the entire checkerboard will be very, very clear to you. Right? So our typical 9:3:3:1, this ratio gets modified to 9:3:4. 9 purple grains, 3 red grains, and 4 white grains. See, so what happens here, this type of interaction is also known as supplementary interaction. Okay, please remember this type of interaction is also known as supplementary interaction. Okay, so if your question is carrying this term supplementary, don't get confused. So why did I say that it is supplementary? See, supplementary genes are the genes which include 2 pairs of non-allelic genes. Here also you can see that 2 pairs of non-allelic genes are here. Both of them are involved in affecting the same character, right? Out of the non-allelic genes, one gene is dominant and can express itself. The second gene is also dominant but expresses only when it is supported by the presence of the first one, right? That is why we call it supplementary gene interactions as well. So I hope it is clear to you. Still, if you have any confusion or any type of doubt, please feel free to get back to me. I will try my best to solve your doubts. And this checkerboard has to be learnt very well. It should be practiced. I'm not telling you mug up, learn it up by heart. No, you should know which gene is controlling which character, which is epistatic to which gene, and you should know how to come to the conclusion, like which is purple, which is red, which is white. You should be very clear with that. Okay. So thank you so much for attending the lecture. Stay tuned for the second lesson, which is also going to be on epistasis. Till then, goodbye.